we've showed that, this is another thing we're working on, and we'll hopefully publish this very soon, that Sordelii produces a protease that actually cleaves VCAM1 and CXCR4. And so we think it's contributing to this profound white cell count. Okay, it's producing something that actually cleaves one of each of these different pairs, the CXCR4 and the VCAM1, which again will likely enhance this dumping, what's taking place with this release of white cells. And so that, along with something else during your reading, when you guys do your research, you'll sit and read tons of manuscripts, trying to get a feel of what's known, how your research fits into what people believe, and then with a skeptical eye, try to convince yourself whether or not to believe whatever this guy just wrote. Okay, and that's how the research world works. And so, doing reading, it has been accepted that granulocyte colony stimulating factor, GCSF, actually stimulates protease release of neutrophils within the bone marrow, which also cleave these receptors, causing this dumping. In fact, bone marrow transplants, you don't do the old school sticking with a needle and recover the bone marrow anymore. What you do now is you administer GCSF to patients, and I think 3 or 4 doses of that creates this dumping, including stem cells. And so you collect blood from this patient, and then isolate these cells and then introduce that to the recipient. And so it makes it much more, it's not as traumatic as it used to be. And it's because of this GCSF, and it's because of the mechanism right here, protease release cleavage, and then the cell release into the blood.
Could you repeat what GCSF?
Oh, sure. It's granulocyte colony stimulating factor. And so, reading that and then looking back at the blood that we collected from the mice infected with Sordelii, we were able to show that at 24 hours and then at 48 hours, we're seeing extremely high levels of GCSF in these mice. So it was making sense to us. Okay, so this must contribute, at least in part, to this leukocyte reaction phenomenon. That's my shifting gears slide. So does that make sense, that the GCSF, and then the protease production by Sordelii itself, all that we think is the reason why we see high white cell counts? Because in difficile, produce basically the same 2 toxins. There's extremely high homology between the lethal toxin and hemorrhagic toxin from Sordelii and the toxin A and B from difficile, almost identical. However, completely different infections. And while a similar symptom, this elevation of white cell count, extremely high in Sordelii and mildly elevated in difficile. And we think what I just showed you is contributing to this extremely high white cell count in the Sordelii patients. Okay?
That spike in itself going to be problematic for you, or is that something you guys are just looking into as kind of a side effect?
Say that again, how the spike?
Is the spike in those white blood cells, is that going to be harmful to the body itself?
Oh, that's a good question. In fact, that's what we say, is the leukomoid reaction simply a phenomenon, or is it actually contributing to these horrible infections? And we don't know the answer to that. I could get some money. We could look into that. That's one of the aims in our grant, is to determine whether or not it's contributing to worse outcome or whether or not it's simply just a marker of infection. Because I don't have the slides, but we do know that these cells, or these white cells circulating, are in an extremely elevated state of activity. They're highly primed, and they're coming out with their knuckles dragging, and they're ready to start a fight, but they can't get to sites of infection. I guess I can show you that here in a second. So they're in circulation, and they can't get out of circulation to go fight the infection, but they're in a highly elevated primed state. And so we don't know. It's kind of like a powder keg or a nitroglycerin state. We think maybe perhaps just a little tap of that, and it's going to release all these toxic superoxide types of radicals that they use to fight infection could be dumped into our circulation, again contributing to worse outcome. We don't know. It's a good question. So kind of shifting gears now, real quickly I want to share with you, we're looking into this lack of innate immune response. So what's going on there? Why aren't people mounting some sort of response to these infections? We did work to show that if you isolate monocytes and lymphocytes from human blood, we do that all the time actually, working with each other. If we need a sample, we'll take turns drawing blood from each other. We'll isolate the white cells, and then we'll use those in vitro in a petri dish, and then expose these cells to different, really whatever you want, toxins, or in this case, what I did was I used Sordelii carcass. I grew it up, and then I killed it. He killed it. So just at the bodies themselves, exposed these monocytes collected from human blood, and showed that these monocytes had a huge TNF-alpha response. TNF-alpha is a pro-inflammatory cytokine. That's kind of what triggers your innate immune response. So we knew that Sordelii was triggering this response in human monocytes. So we knew that the question was whether or not the immune system could even recognize these bugs. We showed that that was in fact the case. They can recognize them. I don't know why I have this slide here. I wish I had shown that earlier. This is our animal model of infection. Again, it mimics what we see in humans. There's the no treatment, and then here's your Sordelii infected, extreme swelling, but there's no real inflammation again. So, and this is in comparison to like a perfringens infection that with these mice, as you can see, severe swelling, but the redness, the inflammatory response that's absent in these Sordelii. So doing more reading, became familiar with the fact that GCSF, that I just explained, used to do these bone marrow transplants, causes this dumping of white cells, is also an anti-inflammatory cytokine. And what it does is it causes release of soluble TNF receptors, which are TNF-alpha antagonists. They'll bind up the TNF-alpha and shut down the innate immune response. So we thought, oh my gosh, this kind of seems to fit with what we're looking at. And so, again, I just showed this slide. This is the GCSF concentrations in animals infected with Sordelii. And then this is the soluble TNF receptor concentrations. Over time, they become extremely elevated. And so as infections get worse, it's pumping more of this stuff out to shut down this inflammatory response. And that's why we believe patients don't display these symptoms during infection. And we just got this stuff, this brand new stuff for us, just published last month. And we also showed that IL-6 was elevated, and IL-6 can be a pro and an anti-inflammatory cytokine, a pleiotropic cytokine. And so we also showed that too. We don't know the role. I think early on it's pro. As you can see, the TNF-alpha, these are three pro-inflammatory cytokines. TNF-alpha and IL-1 beta, and as you can see, they try to elevate early on, and then they just kind of get shut off. And then IL-6 continues to increase, but again, IL-6 can act as an anti-inflammatory cytokine. Okay? So we concluded then that post-production of GCSF seems to be critical in these infections. So if I had money and I really wanted, Sordelii was a big problem around the world, I'd be giving talks around the world about GCSF and blocking GCSF and treating these infections, but unfortunately nobody cares anymore. So we're just doing basic science research now to add to this clinical picture for this pathogen. But nonetheless, it was very exciting for us when we kind of uncovered all these things. I think that's, oh yeah, I just talked to you about this. There's no recommendations in treating these infections. Antibiotics can treat bugs, but there's no way to treat these toxins that are circulating. And so there is a need for antitoxin types of therapy for a lot of Clostridial infections, because it's the only way to really stop the display of these symptoms. Again, you need to stop the actions of these exotoxins and not so much just kill the bugs. Oh yeah, and that's what antivenom is. When you get bit by a rattlesnake, you're targeting the toxin itself.
Is that the way most of the bacteria works? Like most bacterial infections, is it toxins that are released, or is it just?
It depends on the pathogen, right? Depending on the infection, I should say. Again, UTIs, things like that, it's just normal flora getting in the wrong place. But group A Streptococcus, Staph aureus, all those pictures I showed, different viruses producing proteins, these toxins that are, that have evolved over millions of years that can now target specific human proteins that typically or basically interfere with very critical signaling pathways that we need to survive. It will shut them off, and that's when we develop these, nasty infections, or in this case, Sordelii, you start to drain from the inside out and you can't mount fever. HIV, it shuts off the ability of the immune system to communicate, so you can't fight infection in general. So with those pathogens, yes, it's exotoxin mediated. That's right. So any questions with that? I don't know, I already forgot how much time I have, but.
About a little over 15 minutes.
Okay, perfect. So I'll just get through this and we'll just talk. So the other work that I'm doing now, again, trying to transition into the difficile world, is Clostridium difficile. And as we mentioned earlier, the problem with this guy is that it's so resistant to so many antibiotics, and there is a need, a cry now for new antibiotics to treat these infections. It's estimated that approximately 5% of the healthy population carry this organism, and I should have mentioned that earlier. I'd say that all of us carry at least one or two pathogens, as part, and it's intermixed with part of our normal flora, okay? I bet you if we took nasal swabs from everybody in here, I bet you 3 or 4 would have a MRSA, a methicillin-resistant Staph aureus. A few more would probably have a methicillin-sensitive strain, but we all carry these pathogens. It's just that the flora outnumber these pathogens, and so without a critical mass, if you will, of pathogens try to take off and create infection, they lay dormant until the opportunity arises for them to proliferate, okay? And so, again, probably 5% of the normal population carry this bug, but what the biggest problem is that it's the leading cause of infectious diarrhea in hospitals worldwide. They hang out in hospitals, and so if you went in for shoulder surgery and you were on heavy antibiotic to help fight infection, to treat, you're wiping out your flora, and these things are spore formers. Okay, and so they shed, people that have it shed millions of spores every bowel movement. You probably have 10 to 15 bowel movements a day. This is a glove, this is a petri dish, and a nurse was helping a patient with the difficile infection, went to a plate and just pressed his glove on the plate, and they incubated the plate. These are all difficile colonies gained from this guy just working with a patient. I mean, so highly infectious. And again, it's because there's spores everywhere, and so that's, and these people or patients ingest spores, and if you're on antibiotics, and especially one that it's resistant to, well, now you have difficile infection. So now you're going to be in this hospital for twice as long, because not only are we treating shoulder surgery, but we're treating a difficile infection as well, okay? And so it's costing the healthcare community approximately $3 billion in additional healthcare costs a year, because these people in hospitals keep picking it up, and they don't really know how to get rid of these spores. We do know that 10% bleach will wipe out spores, but it's kind of hard to rub that on a patient or give that to a patient. Everything else, these spores are even resistant to normal cleaning solutions that hospitals use to treat wounds. And so, if that person, if you guys can come up with a good idea to develop something to treat or target spores, you're going to be a billionaire. I mean, it's because the world's looking for it. So anyway, so the infections range from mild diarrhea. I guess
I had it
she
in
had,
my 20s.
in her 20s had a difficile infection, but you're young enough. They typically target immunocompromised or older individuals. So if you're younger, I think your immune system's healthy enough to fight the infection. But it's these compromised individuals that develop these nasty infections. Okay, and it's the pseudomembranous colitis. If you look in the colon that has been depleted of its normal flora, these things start to set up shop and grow these little plaques inside there, okay? To the point where they have to basically get rid of the colon. It becomes necrotic. Or it becomes, it's called toxic megacolon, and the swelling is so severe that they have to get rid of this colon as well before it causes death in these patients.
Is that just the colon on A?
Yes, that's just the colon. Yeah, severe swelling, right. And it's because the body's trying to fight this infection. 10 to 15 movements a day, abdominal cramp, really what you would expect with any infection in terms of pain, especially an infection involving the colon. But what's interesting again is this leukocytosis, similar to what we see with Sordellii. But these patients mount fever, so there is a clear difference. The same two toxins cause most of the symptoms associated with these infections, but they're different. different from one another. Okay? One being the fever, bless you. And so I just described this, and this is really the accepted story with difficile infections, especially in the hospital setting. You go in, you get treated for something, whatever the procedure is that you're going through, and then really you're probably exposed to sort of difficile, maybe a non-toxigenic strain to where you're asymptomatic, or you're with a toxigenic strain but you have a healthy immune response to it, so you're asymptomatic, or a toxigenic strain that you don't, your body's unable to mount a response to, and then you develop the symptoms of difficile infection. We were talking about this, I think it was you, before. So a very common therapy now for well, back up. So trying to treat these things is, again, it's become very difficult because of the high resistance. Again, when the talk started, if you expose enough antibiotic to a bug, I don't care what it is, over time it will finally develop a resistance to it. Well, these things hang out in hospitals, so they see antibiotics every day, and so these strains continue to evolve to where they're developing resistance to most antibiotics. And so there's two that have been used over the last, I'd say, 15 years, as vancomycin and metronidazole. I mean, again, if you did a search for difficile antibiotic treatment, that's what you're going to read. 28, let's say, 300 articles describing the use of vancomycin and metronidazole. However, these strains are now developing resistance to these antibiotics. And so newer antibiotics are coming out, this fidaxomicin and tigecycline. Those are ones that we've worked with in our lab. And they've shown strong efficacy with difficile. And so patients, they're very expensive, but it can seem to really help patients that are struggling, again, typically older people, so they don't develop this pseudomembranous colitis or these toxins. So those work, but fecal transplants now are becoming very popular for reasons we just discussed. You got to get the spores out of there. You can treat the bugs, but you can't, it's hard to get rid of the spores. And doing these fecal transplants, wiping everything out that's existing currently in your colon, and then introducing a new flora, I should say, with these transplants, seems to be working very well. Again, it was controversial 10 years ago, but I think it's pretty accepted. The success rate is up there. Did you have a question with that, or did I think we talked about?
I think that was just the.
Oh, the legality, or?
Well, it wasn't so much that, it was what are the alternative methods, besides using the pills?
Antibiotics.
Yeah.
Oh, the antibiotic, yeah.
So
I think that's the major one right now. Again, if you can develop a way an antibody to recognize certain surface markers of the spores, I think that's where it's got to go, to get rid of the spores, to help the immune system clear those out of there. Otherwise, they just lay dormant until you're on antibiotic again, and when things open up, these spores germinate, take off.
So do the spores, my sister's mother-in-law has been going through this for a long time, and she has underlying health issues. But anyway, do the spores, so some of the stuff that I've been reading, sometimes they'll like test the feces and it won't come up positive, or it's like there aren't as many maybe spores, or aren't as many C. diff in her. So she's been going through this for a long time. So are there certain tests that should be done, or can it kind of go strong and then kind of wane a little bit?
Yeah, I think that's, depending on your immune system, I guess, and once you have the infection, right, and then there's assays they can do with your stool to look for toxin A and B production. We have these ELISAs that we use to do all sorts of other stuff, but the hospitals use them. They'll get a fecal sample from a patient, and they'll do a toxin A/B test. Are you guys familiar with PCR? You guys heard of PCR? It's a way to amplify certain regions. Polymerase chain reaction. And so what will happen is you can use that too. You can collect fecal sample, isolate all the DNA in there, and then run these primers to do polymerase chain reaction on certain segments of the toxin, the genes for these toxins. And so they'll identify a patient as positive if you have a positive PCR reaction or if you have a positive ELISA looking for the actual protein of these toxins. But they're so highly sensitive that sometimes I think